Hello, my name is Dr. Roderick L. Roll, and today I will be talking to you about carbohydrates. Carbohydrates are one of the macromolecules that we must consume. In this slide, this was taken from the Food and Drug Administration. This is the food pyramid. How many calories should be consumed, and more importantly, which portion of the calories should be carbohydrates, proteins versus lipids. Carbohydrates should be 56% of your calorie intake. Carbohydrates contain carbon, hydrogen, and oxygen. They contain a specific ratio, 1 carbon to every 2 hydrogen to every 1 oxygen. Or it can be also stated that for every 1 carbon, you have 1 water molecule. Therefore, they call it carbohydrate. Hydrate means water, 1 carbon to 1 water. Examples of carbohydrates are monosaccharides, disaccharides, oligosaccharides, and polysaccharides. We will first talk about monosaccharides, or better yet, better known as simple sugars. In the image below, we have several examples. Glucose is found in sports drinks. Fructose, is found in fruit. Lactose is found in milk, and within lactose, you have a monosaccharide linked to another monosaccharide. Those two are glucose and galactose. In nucleic acids, you have deoxyribose, which is a sugar found in DNA, and ribose, which is found in RNA. A structural isomer of a carbohydrate will form an aldose sugar and a ketose sugar. Previously, we discussed this. The one on the left is an image of glucose, and the one on the right is an image of fructose. The only difference is the position of their carbonyl group. A reducing sugar can donate a free electron from the carbonyl group. So yes, glucose is a reducing sugar. It can donate free electrons. This free electron that glucose and fructose can release will bind to a copper reagent. This interaction is going to form a red precipitate. So this solution that we will use in lab is called the Benedict's reagent. It is a blue liquid. Inside of the blue liquid is the copper reagent. If a reducing sugar is present, and if we had heat to the solution, the reducing sugar will donate a free electron. It will bind to the copper reagent, and it will generate a brick red precipitate. So this is a colorimetric test to help you distinguish what solution has reducing sugars. This is what it would appear like. So again, if you add heat, if you add Benedict's reagent, and inside of the reagent, this blue reagent, you have a copper ion. This copper is going to bind to the free electron that was released from the reducing sugar to form a red color. In this slide, we have some other examples of reducing sugars. Lactose, which is a milk sugar, is a reducing sugar, and so is maltose, versus sucrose is not a reducing sugar. This slide shows you how your experiment should be set up. Everything should be labeled. So we have a distilled water, that's our negative control. We have a reducing sugar, that is our positive control. And we're comparing everything to our positive control. So as you can see, glucose, which is in test tube 3, looks just like reducing sugar. So therefore, glucose is positive for reducing sugar. These are the other 4 tests that we performed. We have potato extract, vegetable oil, egg white, and apple juice. We have, since this experiment, we have added several other examples of solutions that you need to test. So we're talking about the reducing sugars. We're talking about the monosaccharides. So in an aqueous solution, a monosaccharide will form a ring. The first image, the linear structure of glucose, we have labeled all of the carbons, carbon 1 through 6. To the right of that yield sign, we have a ringed form structure of glucose. Again, in an aqueous solution, the monosaccharides will form rings. Be able to distinguish a glucose or monosaccharides in their ring form. Notice that the oxygen is located at 1 o'clock. If you took the abbreviated ring form and superimposed it onto a clock, at the number 1 on the clock would be where the oxygen would be located. At 3 o'clock, that would be carbon 1. You go down to where 5 would be on the clock. That would be carbon 2. And as we keep going clockwise, that would be carbon 3, 4, 5. Carbon 6 would branch off of carbon 6. Carbon 6 would branch off of carbon 5. The next type of carbohydrate we will talk about is a disaccharide. Disaccharides are 2 sugars covalently linked together. In this image, we have taken glucose and fructose, and through a dehydration synthesis reaction, this reaction pulls water out of the reaction and it joins those 2 sugar molecules together. This will form a disaccharide called sucrose. Sucrose will be found in things like honey and sugar cane. Another example of a disaccharide is lactose. Lactose again is milk sugar, and it is composed of galactose and glucose, covalently linked together. The third one we'll talk about is maltose. It is a combination of 2 glucose molecules. This slide shows a dehydration synthesis reaction. You take 2 glucose molecules, you join them together to form maltose, and you release 1 water molecule. The bottom image shows you combining glucose and fructose together. You also release 1 water molecule and you create sucrose. So the first disaccharide we will look at is sucrose. So sucrose is an example of a disaccharide, and it is composed of 2 monomers linked together through a covalent interaction. This glucose and fructose will form honey. Sucrose is also found in sugar cane. The actual molecule sucrose is composed of 50% glucose and 50% fructose. Does high fructose corn syrup cause obesity and disease? Actually, consuming too much high fructose corn syrup causes obesity and disease. The actual sugar itself does not cause these physiological diseases. Consuming too much of it actually causes it. In this slide, I am showing you how glucose is catabolized in the body. Glucose will be catabolized into a molecule called pyruvate. Pyruvate will then go to the mitochondria where it will generate even more ATP or energy. Fructose, which tastes sweeter, can be converted into fructose 6-phosphate, which is one of the intermediates in this glycolysis pathway. So the purpose of showing you this slide is to explain that glucose can be used by the body, and so can fructose. In this slide, I am showing you the historical trends in the consumption of sucrose versus high fructose corn syrup. So the data that we have suggests that the blue squiggly line is refined sugar. That's sucrose from cane sugar or honey. High fructose corn syrup is HFCS. And they have added the instances of obesity. So as you can see, as obesity rates increase, the consumption of refined sugars decreased, and the consumption of high fructose corn syrup increased. So this data suggests that utilizing high fructose corn syrup increases the obesity rate. In this slide, we're comparing cane sugar to high fructose corn syrup. Cane sugar has 50% glucose and 50% fructose. In high fructose corn syrup, you notice that it's a combination of things. In order to make this sugar taste sweeter, you must add extra fructose. So in this corn sugar, we have three components. The first one is HFCS55. That mixture is 55% fructose and 41% glucose. This will therefore make that mixture taste sweeter because of the concentration of fructose in reference to glucose. Also in this corn sugar, you will have a mixture of HFCS42. This will have 42% fructose and 52% glucose. And also in this corn sugar, you have some corn syrup. So collectively, this corn sugar is going to taste sweeter. So again, the question is, does high fructose corn syrup cause obesity and disease? No. Consuming too much of high fructose corn syrup will cause these things. Why have Americans consumed more? The issue is about taste. Because it tastes sweeter, it is more addictive than regular cane sugar. The next disaccharide that we will discuss comes from milk sugars. It is called lactose. Lactose is composed of galactose and glucose. Like I discussed earlier, lactose comes from a cow. It comes from the mammary gland of a pregnant female cow. It does not come from a soy plant. It does not come from a coconut tree. It does not come from an almond plant. This slide shows you the enzyme that some of us no longer produce mass quantities of. Lactase is the enzyme that breaks down lactose into galactose and glucose. Why is milk so important? Milk contains calcium. Osteoporosis is a disease that is caused by the lack of calcium. So drinking or consuming milk that has been fortified with calcium and vitamin D is very vital to you not getting this disease. So osteoporosis is when the bone becomes brittle. So as you can see in this image to the right, that bone has been calcium has been removed from the structural component of the osteon or the osteocyte. In this slide, we're looking at an athlete who has clearly broken his tibia and fibula. Why is this becoming more common? It is becoming more common because these athletes are deficient in calcium. Calcium is stored in the bones. It is also found in this sarcoplasmic reticulum of the muscle fibers in the muscle cell. So when the muscle contracts through the sliding filament theory, calcium is immediately pulled from the sarcoplasmic reticulum. Once that calcium is depleted, it will be retrieved from the bone. So if you don't replenish this calcium that was taken from the bone, then the bone becomes brittle. So make sure you drink milk. Calcium coupled with vitamin D seems to be more beneficial to bone health. So 99% of the body's calcium is stored in the bone and the teeth. We need to consume 1,000 milligrams of calcium per day. Now you can also get calcium from the soy juice and the coconut juice and the almond juice. You just have to make sure it has been fortified with this atom. So we also need about 0.15 milligrams of vitamin D per day. So vitamin D is made by the skin when exposed to adequate sun. And we live in Florida, so this is adequate sun for us. The third disaccharide we will talk about is grain sugar. Grain sugar is composed of two glucose monomers linked together. It is made through an enzymatic activity on starch. So when we are digesting a starch in our mouth, enzymes can hydrolyze dimers off of this polymer. Some examples of this will be seen in malt sugars, beer, and in whiskey. So again, if you look at our image to the bottom right, this is starch. It is a complex polymer. If we start breaking off two sugars that are linked together in pairs, these will be called maltose. These are dimers. These are two glucose monomers linked together. This will be seen in malted sugars. It will also be seen in beer. It will additionally be seen in rice wine called sake. So all of these contain maltose. Sake was originally made through partially digesting starch. So if you chew rice or you chew some kind of potato, a starchy substance, you will then start enzymatic activities in the mouth. If you spit this out and put it in a container, the enzymes in the mouth, amylase, will continue to break down starch. It will break down starch into dimers. These dimers are maltose. The monks used to use this to make the rice wine. The third type of carbohydrate we will talk about is an oligosaccharide. Oligo means a few, about 5 sugar molecules linked together covalently. So this oligosaccharide will be seen linked to a protein that is embedded in the cell membrane. This will make a molecule called a glycoprotein. This glycoprotein can be used as a receptor or a recognition particle. The fourth type that we will discuss is a polysaccharide. Polysaccharide means many sugars linked together. In this image, we're looking at glycogen. These are all examples of a complex carbohydrate. They are also called polysaccharides: starch, cellulose, and glycogen. The first type we will discuss is starch. Starch has two forms, an unbranched form and a branched form. The unbranched form is called amylose. Amylose is a storage sugar for plants. When plants need energy, they can utilize a storage sugar called amylose. The second example is Amylopectin. This is also a storage sugar for plants. The difference is this one is branched. So we have several branches coming off of this main linear form. One is attached to carbon 6 at the top of the image, and another branch is off of carbon 6 on the bottom of this image. This image here shows that the linear form of starch is called amylose and the branched form is called amylopectin. These foods are where starch will be found: potatoes and breads. The next complex carbohydrate, the next polysaccharide we will talk about is chitin. Chitin is a carbohydrate found in the exoskeleton of insects. This slide here shows that chitin can be used as a dissolvable stitch seen in medicine. The next complex carbohydrate is cellulose. Cellulose is a structural carbohydrate found in plants. It helps make up the cell wall of a plant. The next sugar that we will discuss is called glycogen. Glycogen is a branched carbohydrate found as a storage sugar in the liver of humans and in the muscle cells of humans. Now we will discuss a polysaccharide linkage. So these sugars that we've been talking about, when we take monomers and link them together, we can make a dimer or a disaccharide. When we link more together, we can create a oligosaccharide, a few. If we link even more together, we can make a polysaccharide. So let's talk about polysaccharide linkage. We have two categories based on can we break it down. So we have digestible carbohydrates and we have non-digestible carbohydrates. The digestible carbohydrates are on the left. They are linked together chemically through covalent bond interaction. More specifically, it is called an alpha 1,4 glycosidic linkage. The one on the right is showing non-digestible carbohydrates. This is called beta 1,4 glycosidic linkage. So we're going to discuss these extensively. Our dietary consumption should consist of 56% carbohydrates. 180 grams of this should be digestible carbohydrates and 30 grams should be non-digestible carbohydrates. What links these two sugars together, or these many sugars together, are glycosidic linkages. This is nothing more than a covalent bond. To understand the covalent linkage, we need to understand the positioning of the hydroxyl group. If the hydroxyl group is down, we call it alpha linkage. If the hydroxyl group is up, we will call it beta linkage. So in the image below, to the far left, above alpha glucose, look at carbon 1. Carbon 1, the hydroxyl group, the OH, is facing down, so we call that alpha glucose. The one to the right, the OH group, is facing up on carbon 1, so we call it beta linkage. If we look at this disaccharide, maltose, and we look at carbon 1, we notice that the hydroxyl group was facing down. And when it linked to another monosaccharide and formed maltose, carbon 4 on the adjacent sugar linked together. So the two hydroxyl groups were facing down, they linked together, H2O was released, and an oxygen was left. So this is alpha linkage because the hydroxyl group is facing down. If we look at this image, we notice that on carbon 1, the hydroxyl group is facing up. Since it is facing up, this is referred to as beta 1,4 linkage. This will be seen in fiber. Humans are not able to break down fiber because of this beta 1,4 linkage. So in this image, I would like for you to tell me if humans can break this molecule down. Additionally, I would like for you to tell me if humans can break this sugar down. So again, in reference to breaking these sugars down, in reference to polysaccharides or complex carbs, we're looking for the linkage. If it's alpha 1,4 linkage, we have enzymes that can break those molecules down. And if it is a beta 1,4 linkage, we do not currently possess the enzymes to break it down. So therefore, when we consume fiber, fiber is undigestible because of that. Since we're talking about fiber and its undigestible ability, let's go further into fiber. We have soluble fiber versus insoluble fiber. So the soluble fiber will absorb water from its surrounding. So in the large intestines, this is where water absorption takes place. Water will be absorbed out of the large intestines and into the bloodstream, where it will be filtered through the kidneys. So if you have diarrhea, consuming soluble fiber will help with diarrhea. Where will you find soluble fiber? In things such as oats, oatmeal, peas, beans, lentils, and so on. Insoluble fiber will not absorb water. So if you are constipated, you need to consume insoluble fiber. Examples of insoluble fiber is seeds and skins of fruit, whole wheat bread, and so on. This article from the NCBI database is a very interesting article. It talks about cellulose and fiber. Like I stated in the previous slide, we cannot digest fiber. That is not 100% true. Some people have the ability to partially digest fiber. Everybody doesn't have it, but some people do. Therefore, some people are able to acquire energy from fiber, whereas most of us cannot. Also, in lab, you will need to test for the presence of complex carbohydrates like starch. To do this, you will need iodine. Iodine is also called Lugol's solution. When adding a few drops of Lugol or iodine, if starch is present, a blue color will be created. So iodine is an amber or gold color solution. When starch is present, it will turn blue-black to purple. So this is another colorimetric indicator for a macromolecule.